All right, welcome back, everyone. I hope you're doing well. I know that with the coronavirus, things are really starting to pick up in a bad way. So I hope that you're all of you and your friends and family are doing well. I know I'm sort of struggling with the being isolated part myself. Um, so let me know if you need to talk, not just about biochemistry, but if you're just going a little stir crazy, let me know if you'd like. Um, in terms of grades, I'm trying to input everything so that you have a very accurate sensation or whatever of what your grade is as we move into the final. So for last week's in-class activity, if you didn't submit anything, you still can. I'm just putting in zeros whenever we meet deadlines so that you know exactly what your grade is. So I know I've gotten a few additional in-class activities, and that's fine. So please continue to work on those if you haven't. I saw that already a few people submitted for this week's in-class activity. I think after you submit your answer, you should be able to see what others say. So take a look at that. You might find that your answer, you change your mind. And if you have any confusions, let me know. Exam 2, I know I've been saying this for a long time. Now the issue is I have almost all of them graded. I have to manually, using my phone, take pictures of each page and then compile those into PDF, and it's taking forever. I don't have a scanner, and we can't get on campus. So I apologize. My absolute last deadline, if you believe me, will be that I'll get those back to you on Monday. And that should be plenty of time to see if you have any additional questions, because some of that will be on the final. Okay, so where are we headed today? We're done for the most part talking about the nitty-gritties of the coagulation pathway. And I'm working backwards a little bit in terms of I'm now going back and talking about what are the features of signal transduction pathways. And my goal is that you'll be able to compare and contrast at least two signal transduction pathways, the coagulation pathway, as well as those involving G protein coupled receptors and secondary messengers. So today, hopefully today's lecture will be shorter. I'm not going to talk about the nitty-gritty details of the G protein coupled receptors and secondary messengers. That's sections, I believe, 12.2 and 12.3. Today, I'm just talking about section 12.1, where we'll highlight the themes of these pathways. Okay, so if we go back to Twilight and you think about blood, here we were dealing with how blood coagulates, right? But where we're headed next is more along the lines of, that's great, but what does blood taste like? I'm being a little silly, but a lot of it, for example, the G protein coupled receptors are really important for essentially everything, but especially when it comes to sensory, the ability of higher organisms to sense things by sight, by smell, or by taste. By the way, this is not accurate, apparently. It's not as simple. Like, these parts of your tongue don't actually sense just those things in those places. It's more complicated, and yet everyone sort of is familiar with this anyways. So we're focusing on how cellular processes are mediated, involved with sensation, but also other things like smell, sight, hearing. In general, how would you define signal transduction? Like in a very broad sense, using core concepts. It's basically the conversion of information into a chemical change, right? In the blood coagulation pathway, the information was vascular injury, and that had to be converted into a chemical change that the end of this resulted in a chemical change resulting in protein aggregation. So it's kind of confusing in terms of how they describe signal transduction in chapter 12. They describe it as happening only in the context of a cell. And you're going to be dealing, and in this context, you'd be dealing with like a plasma membrane. But I would argue that signal transduction doesn't have to necessarily happen in a cell, because all of this stuff, which is signal transduction, was happening outside of a cell in plasma, right? You didn't actually need a cell to have coagulation. It did involve cells at some point, but it's different from all the signal transduction pathways that are talked about in chapter 12, because there they're talking about propagating a signal from the outside of a cell into the cell. So on one hand, it's kind of semantics. No, you can't, or excuse me, no, it can happen outside of a cell. But at the same time, yes, if you care about biology, signal transduction does involve cells at some point. You could argue that many of the factors that were involved in blood coagulation were made by cells and then secreted. That's true. Overall, since we're made of cells, we generally care about how a signal influences cell behavior. But just to be clear, everything we talked about before isn't really doing this explicitly. It is simply the aggregation of proteins outside of a cell. So we didn't discuss how cell behavior was influenced. However, there are parts of this pathway that we didn't talk about where cell behavior is influenced. So in particular, platelets, right? There are parts of this pathway that would interact with a receptor on platelets that would then lead to another cascade, another signal transduction pathway to be activated that would change how the platelets behave. There's also factors in the blood coagulation pathway that change how immune cells behave, which are really important for activating inflammatory responses. So blood coagulation it's a mix. It does, parts of it don't have to do with cells, but then in the context of making a blood clot, it ends up involving a whole lot of stuff that would be defined more classically, like as the textbook defines it, as signal transduction. So general features of signal transduction are that they are both specific and sensitive. And I want you to be able to distinguish between what is meant by these two words. So sensitivity, and how I'm doing this is sort of like using a clinical example. For example, making a test that determines whether or not, since we're in the midst of this, diagnosing if someone has coronavirus or not, right? If you were to develop an assay that assessed this, there would be two really important parameters that you'd be interested in. One is the sensitivity of that assay, and the other is the specificity. In the context of diagnosing whether or not someone has coronavirus, sensitivity would be the percentage of persons with the disease who are correctly identified by the test, whereas specificity is the percentage of persons without the disease who are correctly excluded by the test. So, for example, let's say a new screening method is developed to diagnose if individuals are infected with coronavirus C+ or not, C-. Let's say we have a sample of 1,000 patients, of which 200 actually have coronavirus and 800 don't. So you could imagine different scenarios. For example, in this first scenario, we have your test correctly identified 100 that had coronavirus, but missed 100 that were also positive. Furthermore, none of the individuals were incorrectly identified as having coronavirus that did not have coronavirus. In other words, was this test sensitive and or specific? It was very specific in that it didn't incorrectly identify people as having coronavirus that did not. So of the 800 that didn't have it, 800 were correctly excluded. So it's very specific. But in terms of sensitivity, we missed 100 people with coronavirus, right? So as we're going through these scenarios, be thinking like, which is best? Which would you prefer? And the answer is sort of, well, it depends. And it's the same for signal transduction. In the second scenario, you've correctly identified all 200 patients that had coronavirus, but you also incorrectly identified 400 as being coronavirus positive that actually were not. So you said 600 people had coronavirus. So in this case, the assay is very sensitive in that you correctly identified everyone, but it's not very specific. It's only 50% specific because 400 of the 800 that did not have coronavirus were not excluded. So this one was highly specific, or excuse me, highly specific. This was highly sensitive. And then you can be somewhere in between. For example, let's say you correctly identified 50 as having coronavirus and incorrectly identified 100 as having coronavirus that did not have coronavirus. Here, the assay is not very sensitive because we've missed 150 individuals with coronavirus, 50 out of 200. And it's somewhat specific, right? We correctly excluded 700 of the patients that didn't have it, but we incorrectly diagnosed 100 of them. They weren't excluded. So if we focus on the first two scenarios, like in scenario one, where the assay was very specific but not sensitive, what would be like the ramifications of that, like in what we're going through right now? The key here is to realize that there are limited resources. And it's the same way in a cell. In scenario one, we were wise in how we spended these limited resources, right? We helped only those who actually had coronavirus. But in the process, we missed some. Some people might needlessly die or be out in the population spreading the disease unknowingly. So the trade-off. In scenario two, because we were very sensitive, but not necessarily specific, we might spend our limited resources unwisely. We waste some of our supplies. We needlessly exhaust our resources on those that do not have coronavirus. But in the process, we helped all of those that had coronavirus be treated and not spread the disease. So the analogy I was trying to make is that the cell is the same way. It has limited resources, and it needs to maximize both of these parameters, both the specificity and sensitivity. The cell is not diagnosing if someone has a disease or not, but it is constantly assessing its environment. So it needs to detect environmental signals and subsequently respond in a coordinated manner. Do you know which animals have the best sense of smell? It's sort of a trick question because apparently it's hard to actually measure this. The way they do it is looking at the number of olfactory receptors in different animals, a part of their brain that's involved in it. So, but it's unclear to know. You would actually have to ask the bear, and to have to like say, yes, I smell that, or no, I don't. But bears, apparently, of those animals studied, have the highest number of these olfactory receptors in a part of their brain. I don't know what Fat Bear Week is about, but it's kind of funny. Where I was going with this analogy is it has important biological consequences, right? The ability of the bear to smell that there's some food in a trash can or a human in a tent many, many miles away helps it greatly. So in this case, and this is actually showing a G protein coupled receptor. If we're dealing with cells specifically and changing the cellular behavior, you're generally speaking going to have involve a receptor in the plasma membrane. And in the case of olfactory sensing, the ligand would be some chemical that's in the air, right? That then binds to a receptor and causes a cell physiological change and subsequent sensing. So G protein coupled receptors, what are they? Well, they're receptors and they're coupled non-covalently to these molecules on the inside of a cell called G proteins. These are proteins that bind to GTP and actually hydrolyze GTP. And we'll talk about that next Monday in a little bit more detail, but they're 7 transmembrane proteins, 7TM. So you'll always see 7 domains that are found in the membrane. So parts of a lot of their side chains are hydrophobic, but then they also have 6 loops, 3 on the inside, 3 on the outside. Okay. Okay, so probably a little bit out of order in how I presented that. I apologize, but what I was trying to do there was I wanted you to understand. I was trying to highlight the difference between specificity, sensitivity in a clinical example, and now the idea here is, okay, how do you get specificity and sensitivity here in a receptor or its downstream components? And this is also true in blood coagulation. What gives rise to specificity in signal transduction? By specificity, we mean that some biological process is proceeding in response to some signal A, but not signal B. And sensitivity would be the biological process proceeds to even a small signal A. Right? So what actually gives rise to these two things? We can start with specificity, where it's, in the clinical example, the percentages of persons who have the disease are correctly identified. This would be like correctly responds to only one type of smell. So what gives rise to the specificity? You can divide it into sort of there are two types of, the answer can be divided into two parts. One is dealing with structure-function relationships. And this has to do with the shape of the molecules. It could be the shape of a particular ligand involved in smell binding to the receptor. The key here is that it's involving weak non-covalent forces and has to do with molecular complementarity. There's also a different aspect of specificity in terms of multicellular organisms. So having multiple cells and different cell types. So just because you have a signal, you don't want every single cell in your body to respond to that signal. You only want those that are important in perhaps in your brain to mediate a response. And so this is getting at the idea that not all receptor genes are expressed in all cells. And even if they are expressed, we also have additional processes such as receptor downregulation or protein trafficking, where the receptor is endocytosin to the cell that changes the number of receptors available to receive a signal. So let's see if you can apply this to the coagulation signaling pathway that we just covered. What gave rise to the specificity of a blood clot forming, of coagulation from proceeding? Do you remember if you can picture or look in your textbook to the actual pathway? The signal was the presentation of a tissue factor that was not accessible to plasma proteins in the absence of vascular damage, but that was made accessible upon vascular damage. So I'm just pointing out to you that there wasn't a receptor per se. Like there wasn't a cell with a receptor in its membrane. But there was that was causing a change in cell, some sort of signaling cascade. But there was a tissue factor that interacted with a protein just floating out in the plasma. And that factor was 7A. So when you had 7A with the transcription factor, or the tissue factor, that's what activated the whole pathway by allowing that first protease to start cleaving all the other proteases. But the specificity comes from molecular complementarity between the tissue factor and 7A. Right? So we didn't actually look at structures of these two things, but their shape was complementary to each other, and their chemistry was complementary to each other. So part of what gives this specificity is also that not only just that 7A binds only to tissue factor through considerations involving its 3D structure and the weak interactions between these two things. It also involved it had to not bind to other things that were already in the plasma. This is not trivial because there are a ton of other potential things in plasma, and so getting at specificity, you need to be able to bind only to tissue factor and not to the other things. So here's from the figure 12-1 in your textbook, what gives rise to signal transduction. Here they're talking about specificity, where a signal molecule fits a binding site on its complementary receptor and others don't. But I'm just pointing out to you that it's not, this isn't in the plasma membrane. 7A is soluble in the plasma. But if we think about this signal cascade, which happens outside of the cell, that's visually shown here. You can also think of it from the flip side, which is that, well, there are components of the blood coagulation that did actually bind with true receptors that were in the plasma membrane of cells and activate what the textbook is calling a signal transduction pathway. One thing we didn't talk about at all is the fact that tissue factor with the protease 7A can actually bind to bona fide G protein coupled receptors. So this actually does bind to a receptor on certain cell types, like, for example, platelets. So here there actually is a true receptor, and the first thing in our coagulation pathway is binding to it. And this is especially important for mediating things like an immune response, activating parts of the inflammatory pathway. And there's also some interesting links to it changing how cancer cells behave. So here's a link, the paper, if you would want to look at it, the link is here, but it's just showing you how in a cancer cell, the tissue factor 7A complex is interacting with this G protein coupled receptor called PAR2. So now we actually have a plasma membrane and a true receptor embedded in the plasma membrane. It's interesting to note that we didn't talk about it explicitly before, but tissue factor itself is embedded in a plasma membrane. But 7A with tissue factor activates PAR2 mediated signal transduction pathways. And there's also interactions with integrins, but we won't really talk about it in the class. Sometimes you'll see these sort of squiggly lines, and that's a hint that you might be looking at a G protein coupled receptor, because there's 1, 2, 3, 4, 5, 6, 7 transmembrane domains. So PAR2 is actually a G protein coupled receptor. Okay, so that was specificity. Or excuse me, was that sensitivity or specificity? We just covered specificity. So this should say sensitivity, and it does. Apologies. This should say sensitivity. Apologies. All right, so sensitivity, again, this is, oh, I apologize, everyone. I'm just going to keep going with it. Okay, so sensitivity. What is sensitivity? It involves amplification. But it also involves a few other factors. So it's not just that the interaction was between your two things was highly specific. The strength of that interaction had to be very high. So Edward and Bella, right, they have an affinity for each other, and it's very high. This is where we get back to other concepts we talked about in the course, such as dissociation constants. So which would you pick to be involved in this aspect, sensitivity of signal transduction, a low or high KD? So, right, KD is a dissociation constant. So if something is binding and staying bound, it's going to have a low KD. So that's the answer, low. So it has a high affinity, a low KD. And just out of curiosity, which of these units might you see? Which would be considered biologically a low KD? Millimolar, micromolar, or nanomolar? So the answer is micromolar. It's usually micromolar or smaller, sometimes even a nanomolar KD. So this right away ensures that the signal is maximally detected. It has high sensitivity because the interaction is so strong. There's a secondary component of, there's usually cooperativity in the binding between a ligand and a receptor. Cooperativity we talked about earlier in the semester in terms of oxygen binding to certain members of the globin family. But here we're talking about occurring between a ligand and a receptor. What do we mean by cooperativity? It involves a change in what? Conformation. So oxygen binding to hemoglobin resulted in a conformational change from a relaxed to a tense state. And that tense state then could bind even stronger to whatever the ligand was. So in this example, it'd be Edward binding to Bella. Bella has a change in shape, and now its affinity is even stronger. And then amplification. So this has to do not just with the initial binding event, but activation of subsequent enzymes in the pathway. So the enzyme is activated by a receptor, which in turn activates a second enzyme, which in turn activates a third enzyme. And the key here is that each time you do this, right, you're like doubling the signal. So initially, Bella got some feelings. Those feelings activated other feelings. And before you know it, you have feelings galore. Or if you prefer the version in your textbook, when an enzyme activates enzymes, the number of affected molecules increases geometrically in an enzyme cascade. Okay. So that's specificity and sensitivity. There are 4 other sort of themes to discuss, which are the modularity of the signal proteins, desensitization, integration, and localized response. So modularity, what we're talking about here is that the proteins involved are binding often to not just one thing, but multiple things. So this protein here in green has multivalent affinities. So it can bind to some signal, but it's also binding to something downstream of it that's important for a response. You can make diverse signaling complexes from interchangeable parts. In terms of these interactions, they're often reversible. So they don't just happen all the time, but addition of post-translationally modifying different components will change the affinities. Most commonly involved is phosphorylation. These domains, they're interacting with other proteins, but there are other things as well. Sometimes they can interact with the cytoskeleton or the plasma membrane. The consequence of interacting with these other things is that parts of the signaling pathway are localized to subcellular parts. So specific places within the cell or specific places on the plasma membrane. You can mix and match different sets of signaling molecules to activate different branches of a signaling pathway. This I'll get into a lot more detail next Monday, but oftentimes you'll see a signaling pathway and you know, it branches out in many different directions. And some of these different directions have different consequences for the cell. So it's important that a single, especially when you're earlier in the pathway, it's important that a single molecule can bind to many different things. Sometimes these interactions are not direct, but indirect. There are scaffold proteins that are important in pathways. They don't themselves have enzymatic activity, but rather they mediate interactions between two enzymes that do have enzymatic activity. So a quick flashback to protein structure-function relationships early in the semester. Given the need for binding multiple partners, which do you think is characteristic of these protein-protein interaction domains or protein lipid interaction domains? Which do you think is characteristic of these domains in signaling molecules? The idea is that they have to be able to bind many things, ordered or disordered. Do you remember there was one specific protein that we talked about that binds like everything? It's really important in cancer. P53. If you got that, congratulations, good job. P53 was very ordered or disordered? It was very disordered. So part of what I'll get to on Monday is one of the challenges. There are at least two challenges in elucidating the structures of proteins involved in signaling pathways. The first is that they are disordered. So it's harder to get crystals that give you structural information. The second is that they are embedded in the plasma membrane in the cell. And when you try and extract them, right, when you make a crystal, it's only the protein. There are some cases where you can crystallize things with the lipid in there, but it's harder to get crystals of. Because of that, these are really tough to see structures of because they're disordered and embedded in lipids. When you try and remove them from the lipids, oftentimes they become insoluble. They crash out of solution, so they make disordered. You can't get structural information from that. Not sure where I was going here. I think just the idea of modularity. So Bella in the movie Twilight, right? She's and signaling components. There's lots of things with arrows pointed at them, and they have to bind things simultaneously. So Bella doesn't have to choose between Edward and Jacob. She can actually interact with both. And if you look at a G protein coupled receptor shown here in purple, that's what this is getting at. You have different domains of your structure that bind to different things. So here at the top, in the extracellular domain of a GPCR, that's what might interact with the ligand, or Edward. And then you can have different domains that interact with the G protein, shown here in blue, right? And this is not just true for receptors, it's also true for all downstream components. So the G protein itself has multiple domains. It's not shown in the cute cartoon analogy, but the G protein interacts with the receptor as well as other G proteins like G beta or G gamma. The next important theme or feature of signal transduction pathways is desensitization or adaptation. The idea here is that the sensitivity of a receptor system is subject to modification. So sensitivity, the ability to exclude signals that are not relevant. right? So Spock is having a response to a signal that's not normal. But in one episode of Star Trek, he does actually cry. Anyways, if you have a signal, you have a response, but the idea of desensitization is that you still have the signal, you still have a ligand binding, but now you don't have a response. If you think about if you go outside for too long and it's really sunny out, and then you come back in, you're actually desensitized to, it takes more light to have the same response. What this involves is the signal binding of some sort of ligand to a receptor not only activates a response, but it actively triggers a negative feedback loop that shuts off the receptor or completely removes it from the receptor through trafficking of the receptor into the cell, endocytic receptor downregulation. Another, we have 2 more to go. Another feature is integration. To some extent, I talked about this when we were talking about really complicated traffic lights. It is not just signal transduction is not just about responding to one signal, but integrating 2 signals. And sometimes those 2 signals can be telling you 2 different things. Shown here in this example, it's talking about the concentration of some secondary messenger, X. Sorry, you have a signal and you want to change either the concentration of a second messenger X or a membrane potential. And the regulatory outcome has to be integrated from input from both receptors. So sometimes this is called crosstalk. So you'll have different branches of a signaling pathway, and they talk to each other. Like by that, we usually mean that there are post-translational modifications happening from one branch of the pathway to the other. And then the last feature is that the response has to be localized. So oftentimes we just see, you'll see a diagram where it's a signal binding to a receptor. But if we actually think about what's happening in the cell, it's usually limited to a physical compartment. So here we have shown lipid rafts. Components of lipid rafts, such as sphingomyelin, they help cluster just certain lipids in certain parts of membranes. The point here is that a lot of times components of the signaling pathway will also be clustered here. Why might this be important? That it be for the signal to be localized at a subcellular level? Well, it's one way to ensure that the message is coordinated with the inside of a cell. So, for example, let's say we have a signal that binds here. Usually you'll have a different component of your signaling complex that is actively destroying some part of your message. The consequence of this is that if you had, it means that your message is only active in this region. If your molecules start diffusing, the message is going to get degraded. So an example of this feature would be like protease inhibitors in the coagulation pathway, right? You had initial activation of these proteases, but what prevented that signal being propagated to all parts of your body? Let's say you had you had a cut on your finger. Why isn't then a blood clot forming in your toe? Well, the answer is because even at the site of your finger, that signal is active, the message is actively being destroyed. In this case, through the activities of a protease inhibitor. So those are the general features of all signaling, signal transducing systems. But then we deal with, you can divide the signal transducers into 4 types. We have your G protein coupled receptors, your receptor enzymes, gated ion channels, and nuclear receptors. The G protein coupled receptors function by indirectly activating enzymes. So here, let's say here's your enzyme in blue. Here's your G protein coupled receptor. The point is that the G protein coupled receptor isn't directly activating this. It's causing the release of some G protein that is now active, and that G protein actually is what activates the enzyme of interest. In the second case, receptor enzyme signaling, so the receptor itself has enzyme activity. And this enzyme activity is on the cytoplasmic side. What activates it is binding of a ligand. It usually leads to some type of conformational change that in turn allows it to be post-translationally modified on the cytoplasmic side, oftentimes phosphorylated. This enzymatic activity is what, of the receptor itself, then can, if it's phosphorylation we're dealing with, can then lead to a kinase cascade where you're post-translationally modifying a bunch of enzymes. This is just split into 2 groups because oftentimes you'll have something that's a factor that goes into the nucleus to cause a change in gene expression. By that meaning turning DNA into RNA to some sort of protein product. The third type is gated ion channels. So here it's not so much that we have an enzymatic change in enzyme function initially, it's just letting something in or out of the cell. The thing of interest is an actual channel in the membrane, not just a receptor. So it causes the movements of ion in or out, and the most obvious, I think, example of this is propagation of an action potential. And then the last one is the nuclear receptor. So this involves the binding of hormones. To actual receptors in the nucleus, right? That are then causing a change in gene expression. So the reason that we're focusing on GPCRs, I guess, is twofold. One is just for practical reasons, we don't have time to talk about all of them. But the second has to do with the fact that GPCRs are everywhere, and they're really fascinating. They have a really wide range in the types of ligand that they bind, ranging from hormones and neurotransmitters to even a photon, which is just kind of crazy. That a photon could be considered a ligand. it's also the target of, ranging on what paper or review you're reading, 30 to 50% of all drugs. So it's very likely that you would deal with G protein coupled receptors, even if it's only indirectly in your future careers. But more importantly, the fact that these are so useful as drugs, it just highlights how ubiquitous they are in cell biology. So this was not one of the most exciting lectures, but we did it. What's next will be actually talking about the G protein coupled receptors, and the idea of what a receptor is. There's some interesting history behind the discovery of G protein coupled receptors that I wanted to talk about. And I also thought I'd point out to you that there was a Nobel Prize awarded in 2012, and it was awarded for the discovery of G protein coupled receptors. So not very long ago. I'm not sure what you all were doing in 2012. I'm guessing you were like in middle school or high school. But if you're interested in getting a preview of part of what I'll talk about next Monday, you can click here. It's the Nobel Prize talk by Robert Lefkowitz. There was also a second person involved in it was a jointly awarded Nobel Prize. But we'll talk about a little bit of the details of mechanisms of G protein coupled receptors. This is, again, sections 12.2 and 12.3 in your textbook. And I just wanted to show this as we end, because, well, 2 people in the class should be really familiar with this figure. It's taken from their presentation. We had someone, Leah and Ayan, talk about retinoid isomerase, which is, I believe, this part of the pathway, going from trans retinol to cis retinol. Do you see any GPCRs, G protein coupled receptors? Well, I'll give it away. Rhodopsin is actually a G protein coupled receptor. And if you listen to this Nobel Prize talk, you'll actually, there's a cool link between rhodopsin and G protein coupled receptors that maybe I'll talk about in the next lecture. Anyways, sort of the big take-home message that I may or may not have been successful in delivering was I wanted you to be able to compare the coagulation signaling pathway with others. And so I thought it was worth describing some of the general features that were actually going on all along in the coagulation pathway. And we'll highlight specific instances of these features being invoked, especially when we talk about G protein coupled receptors. All right, thank you.